Microfluidics is the science of controlling tiny droplets of fluids, often on the scale of nanoliters to picoliters, within small channels. One of the major challenges in microfluidics is ensuring that these droplets remain stable under different conditions. How can droplets be kept intact and predictable? The answer lies in understanding the interplay between surface tension, viscosity, and the design of the microfluidic device. In microfluidic systems, droplets are typically created by flowing two immiscible fluids, such as oil and water, through specially designed channels. The stability of these droplets depends on factors like the fluid's interfacial tension, which helps hold the droplet together, and the viscosity of both fluids, which affects how they flow past one another. For instance, if the surface tension is too low, droplets may merge uncontrollably, while too high a tension might hinder droplet formation altogether. How do researchers manage these delicate balances? By carefully choosing the fluids and additives like surfactants that can modify the interfacial tension to ensure droplets form with uniform size and remain stable during their journey. Environmental conditions such as temperature, pressure, and flow rate also play a critical role. Temperature changes can alter the viscosity of the fluids and the effectiveness of surfactants, leading to instability. In some applications, like drug delivery using lab-on-a-chip devices, maintaining droplet integrity is crucial. For example, a microfluidic device designed to encapsulate a drug within droplets must ensure that these droplets do not coalesce or break apart before reaching their target. How is this achieved? Researchers fine-tune the channel design, fluid properties, and operating conditions to create a robust environment where droplets can travel, merge, or split in a controlled manner. Droplet stability is not only vital for biomedical applications but also for chemical synthesis and environmental monitoring. In chemical reactions performed within droplets, precise control over the droplet's volume and stability can lead to more efficient reactions and easier scaling up of processes. In environmental sensors, stable droplets can be used as microreactors to analyze trace contaminants with high sensitivity. What happens if droplet stability is compromised? Unstable droplets can result in inconsistent reaction conditions, leading to errors in data collection and unreliable outcomes. Moreover, advanced techniques such as the use of external fields, electric or magnetic, can be applied to further control droplet behavior. For example, in electrowetting, an applied voltage can change the wettability of a surface, dynamically altering the shape and movement of droplets. This capability can be particularly useful for applications requiring precise manipulation, such as sorting cells or conducting multiplex chemical reactions. Does the added complexity of these control methods outweigh their benefits? In many cases, the enhanced precision and flexibility they provide justify their implementation in high-end applications. Researchers continue to develop innovative strategies to ensure droplet stability under a variety of conditions. Ongoing studies include computational fluid dynamics simulations and experimental validations to better predict how droplets behave in different microfluidic architectures. Could future developments in material science and microfabrication lead to even more robust droplet systems? Early indications suggest that improvements in channel design and fluid formulation will open new doors for more reliable and efficient microfluidic devices, expanding their applications in healthcare, research, and industry. By understanding and controlling the factors that influence droplet stability, microfluidics can be harnessed for a wide range of applications, from drug delivery systems and chemical reactors to environmental sensors. Maintaining stability under diverse conditions is the key to unlocking the full potential of these tiny yet powerful droplets. The Mpemba effect describes the counterintuitive observation that, under certain conditions, hot water can freeze faster than cold water. This phenomenon has been observed anecdotally for centuries and was formally documented in the 1960s. Researchers have investigated various mechanisms to explain why water at a higher temperature might freeze more quickly than water at a lower temperature. Despite numerous studies, a single universally accepted explanation has not yet been established. In a controlled experiment, A sample of hot water placed in a freezer might reach the freezing point before a sample of cold water under identical conditions. It's like watching a fast runner unexpectedly beat a slower one, challenging our assumptions about starting speed and final outcomes. Historical records show that the Mbemba effect has been noted by scientists and laypeople alike, sparking curiosity and debate over the centuries. Early observers, including ancient scholars, remarked on the phenomenon without fully understanding its causes. This effect has driven scientific inquiry into the fundamental properties of water and phase transitions. It continues to be a subject of rigorous study and critical evaluation in modern thermodynamics. A historical account from the medieval period described how warm water appeared to freeze more quickly than cold water, inspiring later scientific experiments. It's like reading an old mystery novel that hints at an unsolved secret, which later inspires modern detectives to investigate further. One proposed explanation for the Mbemba effect is that hot water evaporates more rapidly, reducing the overall volume that needs to freeze. The loss of water mass through evaporation can accelerate the cooling process. However, while evaporation contributes to the effect, it does not fully account for all observed cases. Critical studies suggest that evaporation is one of several factors influencing the outcome. In an experiment, a container of hot water may lose a significant portion of its mass through evaporation before freezing, thereby freezing faster than a larger volume of cold water. It's like packing a suitcase with fewer items. There's less to carry, making it easier and faster to transport. Convection currents in hot water can lead to a more uniform cooling process, which may contribute to faster freezing. These currents enhance heat transfer throughout the liquid, allowing the water to cool more rapidly. In contrast, colder water may have less vigorous convection, resulting in slower overall cooling. This mechanism works in tandem with other factors to potentially explain the Mbemba effect. When hot water is placed in a freezer, the strong internal convection currents can quickly distribute the heat, leading to an even and faster reduction in temperature. It's like stirring a hot soup to cool it down uniformly, compared to a cold soup that is left unstirred and cools slowly. Supercooling occurs when water cools below its freezing point without turning into ice, and differences in nucleation dynamics may influence the Mbemba effect. Hot water may have fewer impurities and dissolved gases, which can reduce the likelihood of supercooling and promote earlier ice formation. In contrast, colder water might remain in a supercooled state longer if nucleation sites are scarce. This interplay between supercooling and nucleation is a critical area of investigation in understanding the effect. Experiments have shown that hot water sometimes begins to freeze suddenly once a nucleation event occurs, whereas cold water can remain liquid below 0 dC. It's like a seed that suddenly germinates when conditions are just right, while similar seeds may take much longer to sprout if the soil lacks the necessary triggers. The concentration of dissolved gases and impurities in water can affect its freezing behavior by influencing nucleation processes. Hot water, when cooled, tends to release some of these gases, potentially reducing barriers to ice formation. This degassing effect may lower the degree of supercooling and help trigger freezing earlier. However, the influence of dissolved substances is complex and varies with water source and treatment. Water that has been boiled and cooled often freezes more readily than untreated water, partly due to the loss of dissolved gases. It's like cleaning a surface before painting. Removing impurities allows the paint or ice to adhere more uniformly and quickly. The shape and material of the container, as well as ambient conditions like airflow and temperature, can significantly affect the rate of heat loss in water. Different containers may promote varied evaporation rates and convection currents, influencing the freezing process. Environmental factors such as the uniformity of the freezer temperature also play a role in the observed Mbemba effect. Rigorous testing under controlled conditions is necessary to isolate these variables and understand their contributions. In controlled studies, identical volumes of hot and cold water placed in the same type of container can freeze at different rates, depending on subtle differences in container geometry and environmental exposure. It's like baking cookies in different shapes of pans. Each pan can affect how quickly the cookies bake, even with the same recipe and oven temperature. Despite numerous experimental observations, the Mpemba effect remains controversial due to inconsistencies and challenges in reproducibility. Scientific rigor demands that hypotheses are tested under controlled conditions with careful documentation of all variables. Critics argue that without standardized methods, results may vary widely, making the effect difficult to confirm. Ongoing research seeks to clarify these variables and determine whether the effect is universally applicable or context-dependent. Some experiments replicate the Mpemba effect under specific conditions while others fail to observe it, highlighting the need for precise control and reporting of experimental parameters. It's like trying to replicate a complex recipe. If one ingredient or cooking condition varies slightly, the final dish can taste completely different, making consistency crucial. Future investigations into the Mpemba effect aim to develop more comprehensive theoretical models that incorporate factors such as evaporation, convection, supercooling, and dissolved gases. Advanced computational simulations and controlled experiments are essential to tease apart the interplay of these factors, enhancing our understanding of heat transfer and phase transitions in liquids, and contributing to broader fields like thermodynamics and materials science. As experimental techniques improve, scientists hope to resolve the controversies and determine the precise conditions under which the effect occurs. New studies may utilize high-speed cameras and precise temperature sensors to capture the rapid changes in water as it cools and freezes, providing clearer insights into the underlying mechanisms. It's like using a state-of-the-art cooking timer and thermometer to perfect a recipe. More precise tools allow you to understand exactly when and how each ingredient reaches its ideal state.